Are genes your fate? Does nature, genes, or nurture, life experiences and the environment play a greater role in determining your risk of chronic disease? Some people would say, my parents are obese, so I will be obese. Genes are my fate. I've got no hope. No, genes are not your fate. Nurture plays a greater role in determining your risk of chronic disease. In 2003, Duke University oncologist Randy Jertle and one of his postdoctoral students, Robert Waterland, experimented on mice that had the agouti gene, a gene that gives mice yellow coats and a risk of obesity, heart disease, and diabetes. Waterland and Jertle showed that it was possible to silence the gene mutation. The female mice were fed a diet rich in methyl groups. The methyl groups seemed to silence the agouti gene and the offspring were born healthy. Jertle's team fed one group of pregnant mice a diet rich in B vitamins, folic acid and vitamin B12, like green leafy vegetables. Another group, he didn't give any prenatal nutrition. The V vitamins acted like methyl donors. They caused methyl groups to attach more frequently to the agouti gene in utero, thereby altering its expression. And so without altering the genomic structure of the mouse DNA, simply by just giving B vitamins, Jertle and Waterland got agouti mothers to produce healthy brown pups that were of normal weight and not prone to diabetes. This experiment showed that DNA is not your destiny and epigenetics was born. The epigenome controls which genes are expressed and sits on top of the DNA. The epigenome is like an off and on switch, turning genes on and off, and its function can be changed by lifestyle. So the question is, would the same result appear in humans? Brewer found that the environment, nurture, has a significant impact on our genetic legacy. Everything from stress to what we eat to air pollution can alter our genes. Recent studies have demonstrated that a woman's diet during pregnancy could have serious implications to her baby's epigenetic profile. Prenatal diets that are low in nutrients containing methyl groups such as folic acid and vitamin B12 have been linked to asthma and developmental defects in infants. Identical twins can originate from the same DNA, so how come they can turn out so different? For example, one twin could get heart disease at 55 and a twin sister runs marathons in perfect health. The answer is found in nurture, life's experiences and the environment playing a greater role than nature in determining your risk of chronic disease. Epigenetics is the study of how DNA interacts with smaller molecules found within cells that can activate genes and deactivate genes. Consider DNA a recipe book. Those molecules are what determines what gets cooked when. They are not making any decisions themselves. Rather, their presence and concentration within cells make the difference. This is how it works. Genes in DNA are expressed when they are read and transcribed to RNA, which is translated into proteins called ribosomes. Proteins are what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. A common way interference happens is DNA or the proteins it is wrapped around gets labeled with small chemical tags. The set of all the chemical tags that is attached to the genome of a given cell is called the epigenome. Methyl group inhibits the gene expression by derailing the cellular transcription machinery or causing DNA to coil more tightly, making it inaccessible. The gene is still there, but it is silent. Boosting transcription is the opposite. Some chemical tags will unwind the DNA, making it easy to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means it can affect an organism for its entire life. The epigenome mediates a lifelong dialogue between genes and the environment. These epigenetic marks tell your genes to switch on or off, to speak loudly, or to whisper. Environmental factors, nurture, like stress, diet, and prenatal nutrition, can affect genes that is passed from one generation to the next. What you eat or smoke today could affect your health of your children and your grandchildren. Byron studied the conditions in the womb during pregnancy and how it could affect your health, not only as a fetus but well into adulthood. He examined children and grandchildren growing up in Overkirk's Norbotten in feast and famine years. Some years the harvest was bad, so people starved. Some years the crop was abundant and the people gorged themselves. Charles Darwin taught that evolutionary changes take place over many generations and millions of years, but Byron found that through historical evidence that environmental conditions like near death from starvation can somehow leave an imprint on the genetic material in eggs and sperm. These genetic imprints can pass along new traits in a single generation. Byron found that when food was abundant and the kids went from normal eating to gluttony in a single season, they produced sons and grandsons who lived shorter lives. Pembrey also found that smoking by boys in mid-childhood may contribute to obesity in adolescent boys of the next generation. Historical famines help to provide information for the discovery of epigenetic marks that may be modified by the prenatal environment. There was a severe famine during World War II in Holland. If there was excess food in the environment when the paternal grandfather was a 9 to 12-year-old boy, a shortening of the grandchild's survival could be demonstrated, and this could be caused by increased diabetes-associated mortality. If, on the contrary, food was not readily available during the father's slow growth period, then cardiovascular disease mortality of the child as an adult was low. The chemical tags that can turn genes on or off, or speak loudly or whisper, can be influenced by environmental factors including diet, stress, chemical exposure, prenatal nutrition, and medication. It is through epigenetic marks that environmental factors, nurture, can affect the genes that is passed from one generation to the next. Resulting epigenetic changes can lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason genetically identical twins can grow up to have different lives. As twins get older, their epigenome diverge, affecting the way they age and susceptibility to disease. Social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their young, genes in the babies that helped them manage stress were methylated and turned off. Most epigenetic marks were erased when egg and sperm cells were formed, but now some researchers found some of these imprints survive, passing these epigenetic traits into the next generation. Here we see the young rats could not manage stress real well. Your mother's or father's experiences as a child or choices as an adult could actually shape your own epigenome. Even though epigenetic changes are sticky, they are not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may in the long run create a healthy epigenome. Epigenetics could explain aging, cancer origins, heart disease, mental illness, addiction, and many other illnesses. Moshe Seif from McGill University, Montreal, has been a pioneer linking epigenetic changes to the development of disease. Epigenetic patterns are important in the establishment and spread of cancer. Seif has found that common signaling pathways known to lead to cancerous tumours also activate the DNA methylation machinery. Knocking out one of the enzymes in that pathway prevents the tumours from developing. When genes that are typically act to suppress tumours are methylated, the tumours metastasize. Likewise, when genes that typically promote tumour growth are demethylated, that is, the dimmer switches that are normally present are removed, these genes kick into action and cause tumours to grow. Other researchers have identified dozens of genes, all related to the growth and spread of cancer, that become over or undermethylated when the disease gets underway. Fang found in 2003 that the major polyphenol in green tea can prevent deleterious methylation dimmer switching from landing on and shutting down and certain cancer-fighting genes. The researchers described the study as the first to demonstrate that a consumer product can inhibit DNA methylation. Fang and her colleagues have since gone on to show that genistein and other compounds in soy show similar epigenetic effects. A unique feature of nasopharyngeal carcinoma is a strong association with the Epstein-Barr virus. The interplay between the host cell genetics and EBV infection contributes to the development and progression of the nasopharyngeal carcinoma. Epigenetic modifications especially promote methylation is a major mechanism in inactivating the tumours frequently being identified in nasopharyngeal carcinoma. Rappaport in 2016 said, genetic factors are not the major causes of chronic disease. Here's a table showing global deaths attributed to exposure risk factors for chronic disease. Menke found that epigenetics is suggesting that our environment or nurture has a greater influence to play than genetics, nature, when it comes to depression. Morton and colleagues examined the 10-week multimodal wellbeing intervention, or the LIFT project, and reported a reduction in depression and anxiety and improvement in mental health. So, you can see evidence-based strategies from lifestyle medicine and positive psychology can show meaningful improvements in mental health and emotional well-being. Conclusion. Genes are not your fate. DNA is not your destiny. Nurture plays a greater role in determining your risk of chronic disease.